Hello, I am Dharani. My project is about biological microprocessor. We know normal silicon microprocessors working, which we now inculcate those to biological components like DNA, RNA, and proteins for which it can be used in biological computers functioning. In future, we may see small biological computers which are roaming in our bodies, monitoring our health and correcting any problems they find. Biological microprocessors are internally dynamic and possessing their own internal energy systems which are capable of mobility. Researchers from Imperial College London have demonstrated they can build the logic gates which are the building blocks of today's silicon microprocessors out of harmful bugs and chemicals. Logic gates are the fundamental building blocks in silicon circuitry that our entire digital age is based on. Without them, we couldn't process digital information. Intercellular signaling can be used to build logic into biological systems. An interesting aspect lies in compartmentalization of the circuit, where all basic logic gates are implemented in independent single cells that can be cultured together to perform complex logic functions. Such systems are possible in a wide variety of settings, although still a long way off. These biological gates could one day form the building blocks in microscopic biological computers. Here we can see different types of DNA having different color combinations. When they are put through these logic gates, we could get some output with some input. This is the basic thing which happens in a logic gate. What are the biological microprocessors good for? They possess some distinct advantages over silicon-based processors. Biological microprocessors can self-assemble, self-reproduce, and they also have some advantages such as in economic terms also. Moreover, cells can be engineered to sense and respond to environmental signals even under extreme conditions such as high temperature, pressure, and radioactivity. So they also can adapt to new information from a changed environment. From all this, we can see that the main ultimate goals of biocomputing are the monitoring of biological systems. Here, monitoring is stressed because these systems need to be monitored in respect to disease diagnosis or drug screening or to understand some experimental systems, or it can also involve observing the environment. In line with this, a biological computer has been utilized to detect multiple disease indicators such as mRNA of disease related genes associated with small cell lung cancer and prostate cancer. Moreover, they can be used in experimental models such as conditional transgenes or inducible expression systems. And it also includes the control of biological systems. Here, biocomputers can potentially be used to control development, cell differentiation, and reprogramming. All these processes depend upon gene regulatory networks. Metabolic engineering has the potential to produce from simple, inexpensive starting materials a large number of chemicals that are currently derived from non-renewable resources or limited natural resources. So, the metabolic flux can potentially be controlled by a biocomputer. Interesting thing is that it can also be used to control the immune system by a biological computer, as in transplantation medicine. What are the building blocks of a biological microprocessor? Like the normal silicon microprocessor, it also contains input unit, ALU unit, control unit, and memory unit. Now, let's see in detail what each unit is consisting of. In the input-output unit, a biomolecular input-output device is basically an engineering set of chemical reaction with input and output molecules with distinct concentrations formalized. As an example, for the case of the two input devices, here we see input molecule 1 and input molecule 2, which upon combining gives this output molecule. Here we have 1 and 0 representing some concentration regions in which the molecule is active. Here we can see there are increased concentration regions and the time is also increasing in the x-axis. If we see clearly, this is the off region of these molecules and this is the on region of this molecule. Upon changing concentrations, we can see how these are varying. And this this image shows a switch which produces an on and an off state. This figure gives an example of a switch in a synthetic gene network. Off is a state where no detectable EGFP expression is seen, and LACL repressor proteins, which are constitutively expressed, bind to introns with the lac operator sites, inducing transcriptional repression of EGFP and TETR respectively. Repression of TETR allows transcription of shRNA, which can subsequently bind into its target sequence and repress its shRNA's target. And this is what happens in a input-output unit when we consider these type of molecules. But different types of molecules produce different kinds of input and output, like the chemical reactions. Now let's see how ALU unit works in a biological microprocessor. We know that in a silicon microprocessor, ALU unit is responsible for arithmetic operations as well as the logical operations. By using the best use of logic gates, uh, we perform these. But when it comes to biological microprocessor, we need to use the logic gates which I've discussed before, by using certain components and certain chemical reactions that will lead to desired results. Biological computers have shown to be able to execute simple arithmetic such as addition and subtraction. A combination of several DNA hybridization based logic gates make it possible to calculate an integral part of square root of a 4-bit binary number. In a similar way, logical operations can also be done by using these logic gates. For example, a deoxyribose based automation is able to play a complete game of tic-tac-toe. Can you imagine that? A device based on DNA recombination was able to solve a sorting problem where a stack of distinct objects needed to be placed in a certain order and orientation using the minimum number of manipulations. And there is also an astonishing fact that a molecular algorithm based on ribonuclease digestion to manipulate strands of a 10-bit binary RNA library has been used to address the so-called knight problem, which asks the what configurations of knights in a chess game can one place on a 3x3 chessboard, not 8x8, such that no knight is attacking any other knight on the board. So such kind of problems can be solved by using biological components. Now let's see how the logic gates are built in the ALU system. AND gate is based on a transcriptor T and a RNA polymerase. Um, Here 1 means input A and B is sensed or output is released, where 0 means not. And AND gate can be built on a transcriptor T, an asymmetric transcription terminator which can block RNA polymerase flows in one direction. If both the terminators are flipped, induced by their respective input signals A and B, RNA polymerase flows unhindered. And similarly, we can see a NOT gate, another type of Boolean. A NOT gate can be built on a DNA-based catalyst. The deoxyribose is in active form if no input is present. A cleavage activity results in this case with a fluorescent oligonucleotide F as an output. An oligonucleotide input 1 leads the hybridization of the input strand, which is before green in color, with the closed loop strand such that which is marked purple. This results in an open loop and the absence of fluorescent product, and it is released outside. Now let us see how a NOR gate is built. NOR gate is generally a combination of an OR gate followed by a NOT gate. Uh, Two RNA aptamers translates GFP only in the absence of inputs like a NOR inputs like in a NOR gate. You generally have a NOR gate as if you don't have both the inputs, then only the input will be one. Um, Here we have a sensor made up of RNA aptamer. The actual component made of hammered ribosome, which is purple in color. The coupling sequence between these parts is blue in color. The translation of the gene of interest, here we have GFP. GFP is nothing but green fluorescent protein enclosed upstream of the device, doesn't occur in the presence of the other inputs. Uh, it is only possible in the absence of the inputs A and B. This is another type of the gate which we know, XOR gate. Here we will get the input either if A or B only is present, then only we can get the output. This system is built from Escherichia coli colonies, whereas each colony consists of a strain engineered to contain a single gate. The three colonies cells are 1, 2, 3 containing a NOR gate, and the fourth cell is containing a buffer gate. Buffer gate is nothing but two NOT gates uh, which are in a series. We can see here, right, when the inputs A and B are absent or present in both of them, no output is seen. But if either A or B is present, then a yellow color fluorescent protein is uh, seen outside. So now let's go to the control unit. Control unit is also the main component of the CPU in the silicon microprocessor. Similarly, here, state machines are used to know about the transitions that are being taken place in the CPU. State machines have been engineered with biomolecules in our case. The first experimental implementation of finite state machine comprising of DNA and DNA manipulating enzymes was published by Benson et al. In 2001. How to build a finite state machines using biological parts. We see here symbols A and B are written on a tape which is read by the machine later, read by letter from left to right. In this example, the tape ends with the final letter B. Each letter provides the instruction to the machine into which the states S1 and S0 can be reached. The machine should move here, means move to state 0 or S0, and if a B comes, then move to state 1. If another B comes, be in the state S1. If another A comes, turn around in S0. So this is the basic thing which happens in a state machine.